So today I'm really pleased to introduce to you Christoph Meyer, who's going to be talking about bats in fragmented tropical landscapes, insight from Amazonian fragmentation experiments. Christoph carries out research that focuses on assessing species responses to habitat fragmentations and land use change. He also focuses on elucidating their mechanistic basis underlying patterns of species distributions in human-modified landscapes. Christoph's research focuses mainly on the tropics and using bats as model groups. Christoph holds a MSc in biology from the University of Würzburg in Germany and a PhD in ecology from Ulm University in Germany as well. Christoph did postdocs at the University of Ulm and the University of Lisbon in Portugal before joining the University of Salford in the UK as a lecturer in Global Ecology and Conservation. I'm really interested in hearing Christoph's exciting work on bat conservation and in fragmented tropical landscape. So Christoph, I'm going to stop sharing now and I'll let you share your screen. And I'm going to ask everyone just before we start to please make sure you turn off your microphone while we're talking. So make sure you mute yourself just so we don't have feedback.
Okay, thanks very much, Orly, for the nice introduction. Welcome, everybody, and also many thanks to Rachel for inviting me to give this webinar today. Okay, so yes, I will try in today's webinar basically to give you a bit of an overview of the kind of research that we've been doing in my research group over the roughly last 10 years or so. And where we've been studying bat responses to fragmented landscapes, specifically making use of a big fragmentation experiment in the Brazilian Amazon. And just a bit of introductory background, really. Land use change, as you all know, is one of the key drivers of the global biodiversity crisis that we're currently experiencing. As an example here, over 150 million hectares of tropical forest, for instance, have been converted for agricultural purposes between the 1980s and 2012 alone. Deforestation continues to be rampant across many tropical countries, specifically in Brazil, in the Brazilian Amazon, while for quite a while we can see that there were some positive signs in terms of deforestation rates declining roughly after 2002. We unfortunately now are already in an upward trajectory again. Also, a lot of that has to do with the current government of Bolsonaro that is obviously very much opposed to driving deforestation rates down and is rather investing quite heavily in exploiting the Amazon as much as possible. So the Brazilian Amazon alone has lost already 20% of its original forest cover, and certainly, deforestation rates in that region particularly are projected to continue to be very high in the foreseeable future. So anywhere between 23 and 48 million hectares are likely to be lost additionally by the year 2030. Now, obviously, this land use change and concomitant deforestation has resulted in vast areas really of fragmented, heavily fragmented landscapes. And what we see in a lot of areas, specifically, let me see if I get this little animation here to work. If it doesn't, no problem. What it shows basically is a very common pattern of fragmentation, often referred to as the fishbone pattern of fragmentation. That's very common in the southeastern region of the Amazon, the so-called arc of deforestation. A lot of that is basically caused by road construction, lots of primary roads and then smaller side roads going off of that, which create this fishbone pattern of forest fragmentation. So we're left with these heavily fragmented landscapes, which are obviously now a very ubiquitous feature of human modified landscapes across the tropics, not only in Brazil, of course. So in the Brazilian Amazon specifically, an important driver of deforestation always has been and continues to be, one has to say, the conversion of natural forest habitat into cattle pasture. So here, of the different drivers of deforestation in the Brazilian Amazon, really conversion into pasture accounts for roughly 63% here. And considerable fractions really of the Brazilian Amazon, roughly 12%, have been converted into cattle pastures. Now, what often happens in this context, though, is that these cattle ranches, they are not very productive in the long run. So a lot of them actually lose their economic viability after a few years, and a large proportion of these areas that were cleared for pasture land are actually abandoned after a few years, allowing forests to regenerate. And in fact, what we're seeing not only in the Brazilian Amazon, but also in a lot of other tropical regions, is that regenerating secondary forests following these larger scale shifts in human settlement and also agricultural activities, these are actually leading to a rise of regenerating forests across the tropics. So secondary forests now really, they constitute pretty large areas in terms of tropical forest cover, for instance, 22% in the Brazilian Amazon. So a question then that is very important to ask, obviously, what is the capacity of secondary forests to preserve species and to buffer the impacts of fragmentation on assemblages in human modified tropical landscapes? So this is something that we've been interested in addressing at the Biological Dynamics of Forest Fragments project, which is located right in the heart of the Brazilian Amazon, and also in the heart of what is clearly a world bat biodiversity hotspot. So if you look at this map here very clearly, we embarked on a mission here where we had to excitingly deal with lots of different bat species, but more about that in a few minutes. So Amazonian bats in general, they are highly diverse. We find over 150 species alone to co-occur in the Brazilian Amazon. The dominant bat family is the New World leaf-nosed bats, the Phyllostomidea. In addition to that, we find 8 other bat families. Obviously, here just a few illustrations highlighting some of the enormous diversity really that we find. So obviously very exciting. If you study bats in the UK, it's hard to see the same level of morphological and also ecological diversity, obviously. So bats there fulfill, as in all tropical ecosystems, important ecological functions as pollinators, seed dispersers, and also in terms of arthropod suppression. Now, a bit about the study area. As I said, the Biological Dynamics of Forest Fragments project, or usually referred to as BDFFP, it's a project that goes back for several decades now already. It was basically initiated by Thomas Lovejoy, who has been in the news, unfortunately, fairly recently, because he sadly passed away on Christmas last year. He was really one of the great Amazonian conservationists, and he was the one who initiated this particular project in the late 1970s. And it's basically been the world's longest and largest, longest running and also largest experimental study in terms of areal extent that really looked at the effects of studying the effects of habitat fragmentation. And basically what happened back then, forest fragments of various sizes, 1, 10, and 100 hectares in size, were created to isolate, were created to basically create cattle pastures. And so in addition to that, the project also established a range of control plots in very nearby continuous forest. And initially, the project's specific aims were mostly to look at actually the effects of fragment area on how different groups of animals, for instance, or rainforest biota in general, respond to area effects. But over the decades, really, this has developed into a much larger project that has also broadened its focus considerably to look at the wider dynamics of the landscape, to study aspects in relation to forest regeneration in the matrix around those fragments, and also to look at regional and global change phenomena, importantly, also climate change issues. And here in that publication shown here, this is basically kind of the latest summary of the findings of this huge project, really. Importantly, this is a landscape where basically happened what I just described before, is those cattle ranches around the forest fragments, they very quickly became unproductive and ended up being abandoned. So this is kind of what the matrix looked like initially, and then roughly 15 years after fragment isolation, and roughly 30 years after fragment isolation, which is kind of when the time period when we started to work at the BDFFP. And specifically, I want to just present here a few of the key findings. I can't unfortunately really go into every study that we've done, but I'm just going to focus on some of the key findings here, really. A key issue that we wanted to tackle in this research project was really to look at how did the forest regeneration in the matrix around those fragments, how did that influence the bat community across this specific landscape? And we did so basically by looking at different diversity dimensions. We kind of looked at this from the more traditional angle of using mostly taxonomic diversity to evaluate species response, but we also added a functional diversity, phylogenetic diversity component to complement and to get this and to get a complete as possible a picture of how bats respond across this landscape, and importantly, how did this regeneration of secondary forest here in the matrix around those fragments, how did that influence these three diversity facets? So when we talk about taxonomic diversity, we're obviously referring primarily to species richness and relative abundances. For functional diversity, we looked at a number of species traits of bats that are linked to ecosystem functioning and services. And finally, in terms of phylogenetic diversity, this kind of captures the evolutionary history of a set of species. So here are some results. I know Ricardo and Fabio seem to be in the audience, and they would probably be much better placed than myself to present these results. So Ricardo Rocha, he did his PhD mostly looking at the taxonomic diversity aspects in relation to phyllostomid bats, and Fabio Farneda, he looked mostly at functional and phylogenetic diversity. And basically, we managed to get quite a bit of data together. The important thing here is that we managed to capitalize on data that had been collected previously by Erika Sampaio, a former PhD student of Elizabeth Kalko in Germany. She was the first one to work on bats at the BDFFP, and that was roughly 15 years after initial fragment creation, so in the late 90s. And our Brazilian collaborator, Paulo Bobrovic, he sampled the secondary forest matrix. So these sites basically served as comparison here. And so for the specific temporal comparison, we looked at 6 forest fragments, 6 continuous forest control sites, and 7 sampling sites in the matrix, where we sampled with mist nets. And then, between 2011 and 14, we basically resampled those sites using the same methodology. That resulted in quite a number of bats that were captured from over 50 species. So mostly phylostomids, and also we included here Pteronotus as a more morbid species, because it can be also well sampled with mist nets. And so here's some of the main findings, really. In terms of the taxonomic diversity component, what we observed is when we divided bat species into habitat generalists and habitat specialists, we found that these groups, they really showed contrasting responses here. When you look at here, you've got a plot that shows the percentage of species that increased, shown here as the green bars, or decreased in the different habitat types: continuous forest, forest fragments, and the secondary forest matrix. So what we found here is that basically these old growth specialist species, and these are mostly comprised of the gleaning animalivorous bat species, essentially, these really show increases across all habitats, both in occupancy and in abundance. In case of the generalists, though, we found that especially in the secondary forest, that these generalist species, they tended to decline quite substantially between those 2 time periods. Okay, so we got this interesting contrasting response of those different groups. Now, in terms of the functional diversity response, we found quite an interesting recovery here, or functional alpha diversity, especially here in secondary forest. Okay. Note, however, that we're still quite far away here from a recovery to levels observed in continuous forest. Okay, so that's quite important to note. Quite important to note. In terms of beta diversity, we also looked at functional beta diversity changes, and what we found here, again, these were most prominent in secondary forest. And when we partitioned this beta diversity into its richness component and replacement component, we found that really trait gains here were mostly responsible for these pronounced beta diversity changes in secondary forest. We also looked, as I said, at the phylogenetic diversity component, and what we observed here is similar to what we found for functional alpha diversity. Also, a recovery of phylogenetic diversity in secondary forest, and in this case, even what seems to be quite a complete recovery to levels observed in continuous forest. And that is also reflected in the fact that extinction probabilities of certain species, lineages, importantly, of gleaning animalivorous species, they were reduced in late stage secondary forest. So in our second sampling period, we certainly had this very clear positive effect on those animalivorous gleaning species. Now, apart from these temporal effects, we were also interested in obviously looking at these more general fragmentation effects in relation to fragment area, in relation to edge effects, and general matrix effects. So these questions we were basically able to address using a larger number of sites, 9 continuous forest sites here, 8 fragments and 8 matrix sites, and continuous forest sites and fragments were sampled at the interior and also at their edges, and then we had the matrix sites that were in the secondary forest around those fragments. So also here a few results in relation to that. What were key findings here? Well, first of all, we found that that importantly the smaller fragments, the edge sites, and or regrowth sites in the matrix, they tended to harbor really fewer species than continuous forest. That can clearly be seen here. Quite a complex graph. There's quite a bit of information in here, but really what is very clear here, that especially the 1 hectare fragments, they are clearly much less species rich than continuous forest sites. Now, when we looked at abundance patterns, again, we found and looked here and teased things apart a bit by functional group or ensemble. We found that frugivorous species here showed, again, contrasting patterns to gleaning animalivorous species. So very clearly here, comparing continuous forests and fragment sites, gleaning animalivorous species basically had lower capture rates, lower relative abundances in fragments compared to continuous forest, whereas frugivores actually, they tended to do quite well, especially when we look here at the matrix site. So there is still probably able to make use of these earlier successional shrub resources that were found in the matrix. Also, we looked at this from a phylogenetic perspective, phylogenetic diversity perspective, and clearly found here evidence for phylogenetic erosion in these small fragments. Particularly, again, these 1 hectare fragments are really depauperate when it comes to phylogenetic diversity. So there's a lot of evolutionary history lost basically in those assemblages that inhabit these smaller 1 hectare fragments, and that's an outcome of a master thesis done by Sabrina Aninta, who looked at this specific aspect. Now, you may wonder, what about all these other families of bats, which I haven't talked about yet? So all these previous results were for phyllostomid bats, which, yes, they make up a big chunk, obviously, of the bat fauna at the BDFFP. But we obviously also wanted to know a bit about what's going on with the aerial insectivorous species. So for that, another former PhD student of mine, Adrià López Balcells, not sure if he's here as well, he collected and put together a tremendous amount really of field data collected over again many years, over 1 million recordings basically. So he and at the moment also another master's student of mine, Sarah Rowley, they explored what is the again taxonomic, functional, and phylogenetic diversity response of aerial insectivorous bat species in the VDFFB landscape. So comparing again here across the different habitat types comprised of these different sized fragments, and looking also at this comparison of interior habitat versus edge habitat versus the secondary forest matrix. So an important finding here was that basically, again, these smaller fragments, in particular, again, the 1 hectare fragments, but also the 10 hectare fragments. So basically the secondary forest adjacent to those smallest fragment size classes, they basically showed a clear reduction in all 3 diversity dimensions. In addition, we also found here that that in fact the largest fragment size class, the 100 hectare fragments, they have levels of taxonomic, functional, and phylogenetic diversity that is in fact comparable, similar in some cases, particularly for phylogenetic diversity, even somewhat higher than what we observed in continuous forest. And also looking specifically again at edge effects, here we found actually aerial insectivorous bats, they reach a very high diversity at the primary secondary forest interface. As you can see here, we find here quite a positive response again for all 3 diversity facets at the edges, and so this is certainly an interesting finding. Juliana Appel, who did her PhD also using some of the data of that Adrià collected, and I think she's also in the audience. She was interested in looking at what's going on with the aerial insectivorous bats from a more species-specific perspective, and not only looking at habitat disturbance effects, but also including other variables that we know are quite important modulators of the activity of aerial insectivorous bats. Importantly, she was interested in also looking at the effects of moonlight and how that interacted with the habitat. And in fact, what she found, though, is that very clearly, habitat disturbance here clearly has a greater effect on the majority of species. Clearly, for this set of species for which we had sufficient data to analyze them at the specific species level, she clearly found that the secondary forest matrix still doesn't seem to hold the same foraging opportunities for many of those species. Only for a few of them, here the mormopits, they in fact we found higher activity for secondary forest, but for 6 of these other species here, actually activity was reduced in secondary forest still. And moonlight, in fact, played only a rather limited role in explaining activity patterns in this landscape. Another important aspect that we investigated, again using multiple data sets in this case. I'm going to be presenting the results first again for phylostomates and then for we also more recently also looked at this for aerial insectivorous bats, in fact. But the question really was to get a bit of a better understanding. What's the relative importance of local habitat structure, vegetation structure, and landscape structure in determining bat species diversity and abundance? So we looked at measures of local habitat quality. We basically quantified various variables describing vegetation structure, and at the landscape scale, we looked at forest cover as a measure of landscape composition, and also looked at several configurational metrics to describe the overall landscape structure. So these are metrics that we considered, and given that it had been shown that bat species respond very often in a scale-specific manner to landscape metrics, we calculated those at several spatial scales. And I just want to show you here some of the key findings. At assemblage level, we looked again here at a number of metrics: species richness, evenness, and abundance. And this rather complex figure basically summarizes, first of all, the spatial scale on the y-axis, and then the different variables, predictive variables that we're interested in: vegetation structure, forest cover, edge density, and patch density. And the bubble size here basically stands for the amount of variation explained in our statistical model, and then we've got black and white for positive and negative effects, basically. So what we found is, first of all, yes, there seem to be scale-dependent responses very clearly, and overall, we found that there were positive relationships of vegetation structure with all the three different response metrics: species richness, evenness, and also abundance. Forest cover was the most important predictor of species richness, as you can clearly see here, by far the largest bubbles or circles, and also for species evenness. So there was a positive correlation here between those response variables and forest cover in the landscape, whereas for abundance, we found a negative relationship. Now, interestingly, when we then decided to dig a bit deeper, again, this was work led by Ricardo Rocha, and so we split things up a bit further, looking into also seasonal effects and also gender effects. And so when we analyzed basically now the data at species level, we also found that really these associations with local vegetation structure and various landscape metrics, they really seem to be season and also gender specific, probably reflecting obviously the different resource requirements also of female bats and male bats, and when resources are more available across the landscape. So unfortunately, we were only able to really run this kind of analysis for very few bat species, but this was quite an interesting finding, that really we have kind of this very interesting response here, not only to season but also variation here in between males and females. As I said, we also looked at this more recently for aerial insectivorous bats, and only found very subtle responses here for functional diversity. No clear associations here with either taxonomic or phylogenetic diversity, and measures of vegetation structure or any of the landscape metrics. However, forest cover had a positive effect here for functional diversity, and edge density and patch density had negative effects respectively, but only at certain spatial scales. So Interestingly, that, you know, obviously what we kind of expected is to find weaker responses compared to the phyllostomid species, especially in relation to vegetation structure. So it kind of makes sense that you know, aerial insectivorous bats, a lot of those, obviously molluscids and so on, that will forage over the canopy, so that they wouldn't be maybe too responding to vegetation structure. So, but it's still interesting to see how this particular ensemble shows certain responses at certain spatial scales here to some of these landscape metrics. So kind of as a few key take-home messages really, habitat specialists, as we found, they basically had an overall positive effect here that we found of the regeneration of the secondary forest in the matrix on their patterns of occupancy and abundance across all the habitat types that we studied. And likely that's going to be a reflection here of the diversity of food resources in the matrix that likely increased as the matrix regenerated over time. And this matrix regeneration will obviously also have increased the habitat suitability for these gleaning animalivorous bat species, these more old growth forest specialist species, by decreasing or lowering the magnitude of edge effects. And that's clearly also highlighted by some of the findings on, for instance, on the phylogenetic diversity perspective that was presented before. The habitat generalists here, what we found, if you recall, that the temporal effects here were negligible for forest fragments, and they decreased in secondary forest, likely as a result of the decrease in the availability of their preferred food resources. So a lot of these pioneer plants, which were very abundant in the early in the first study period in the late 90s, they kind of disappeared, and that reduced availability of, for instance, important foraging plants such as piper and vismia. They will probably have translated into this negative response in secondary forest. We also, as I showed you, hopefully, is that roughly 30 years of secondary forest regeneration seem to have been sufficient for the partial recovery of functional diversity and the full recovery of phylogenetic diversity in the BDFFP matrix. And in general, I think this adds to a growing body of evidence from the BDFFP, also where other taxonomic groups such as birds have been studied in relation to similar aspects, and also from other study sites, really, that these regenerating forests across the tropics are really act as important repositories of functional and phylogenetic diversity for bats. But as I said, also studies have demonstrated similar patterns for other sites. Now, in the same, what we can basically then say is it's very important that we restore these degraded matrix habitats that of which obviously there still exist very many in human modified landscapes, and ways to do so are numerous. It can be either via natural regeneration, active regeneration, so tree planting, or human assisted regeneration, or a combination of those. It's really all about managing adequately these degraded matrix habitats if we want to maximize the conservation value and potential of these regenerating secondary forests. And importantly, we need to make sure that they're protected in the long term. A lot of studies have shown that the half-life of secondary forest is fairly short, and in many cases, after a few years already, a secondary forest will be cut down again. So it's really all about making sure that we guarantee that they are around for a sufficiently long enough time to make sure that we can maximize the conservation potential. However, what our research also clearly showed is secondary forests are still no substitute for continuous primary forest. Okay, there still are these old growth specialists, and even after all these decades of forest regeneration, we still observed differences between those secondary forests and continuous forests, and that obviously means that we also need to heavily invest into protecting remnants of primary forest. So these should be basically complementary strategies if we want to really maximize bat diversity and fragmented tropical landscapes. And I think that is something that one can generalize. Also what our study clearly showed, our studies clearly showed, was that area and edge effects are even noticeable in a landscape such as the BDFFP landscape, where we have a low contrast, low structural contrast between those forest fragments and the matrix. So another aspect that obviously was very clear here, that biodiversity erosion in specifically the smallest fragments, the 1 hectare fragments, was quite prominent, and not only manifested as species loss, but also as the loss of overall evolutionary history, loss of ecological functions, and that is the latter is obviously likely going to result in also reduced ecosystem service provisioning by bats. And also this comparison of the importance of local versus landscape scale characteristics. It seems to be clear here that both play a certain role in explaining how bats respond across this fragmented landscapes, and so certainly management efforts should kind of have to tailor for both, and should focus on both local habitat preservation and also landscape scale habitat preservation. I would just want to finish by highlighting a few other things that happened, as started maybe as side projects. So Adria, he basically embarked on this huge adventure of putting together a field guide to the bats of the Amazon. And so this started maybe as a small side project, but developed into really a fascinating project outcome. So in 2018 was published the field guide to the bats of the Amazon. A fantastic piece of work, and hopefully useful resource to a lot of people who want to continue to work in the Amazon. And in addition to obviously having morphological keys, what you would normally expect from a field guide, it also includes the first echolocation keys for Amazonian bats. We've made lots of new discoveries along the way as part of this larger project, ranging from descriptions of the echolocation calls of several species that remained not described certainly, or other aspects such as range expansions for certain species. So quite a number of also natural history observations. A former master student of mine, Natalie Jos, she also led a paper recently that looked at the call design and call variation across phyllostomid bats, and obviously usually there's been a lot of focus in terms of echolocation research on aerial insectivorous bats. So this was also an interesting and valuable outcome of this project, that we learned a bit more about really call variation in Amazonian bats. As part of Adria's research, we also gained insights into how can we best optimize acoustic surveys. So we contrasted, for instance, here, looked at aspects of what is the minimum sampling effort required to achieve a certain level of inventory completeness. And he contrasted that under two different landscape scenarios. Basically, an aspect I did not touch upon here was that while at the end of our data collection, basically those BDFFP fragments, they were re-isolated again through the clearing of a 100-meter wide vegetation strip around each fragment. And so we also looked at the shorter term effects of this abrupt change in matrix composition. And so that was one aspect that Adria also evaluated here in this study, looking how does this landscape, this change in landscape context, how does that influence and impact on our on the sampling effort that we need to invest to basically inventory aerial insectivorous bat species. And he also obviously was faced with the tremendous task of basically manually identifying species in thousands and thousands and thousands of calls. So he came up with a nice idea of how can we combine automatic species classifiers while at the same time still applying manual post-validation to make sure that we end up with reliable species identifications. However, have at least done what we can in terms of minimizing the workload associated with going all these calls. So that was a rather short overview of 10 years of research, basically. I think, first of all, many thanks to all former PhD students, master students involved in the project, project collaborators, of which there were many, the BDFFP management team, all the field assistants, funding organizations. Many thanks to Adria and Aurel Massana for the fantastic photos, without which this presentation would have been a lot duller. Hopefully you liked some of the pictures. And many thanks to you for listening, and I'll be happy to take any questions.
Thank you, Christoph, for a great talk. It's amazing to have such a long-term experiments running on that can really provide us with proper insights about what's happening there and the impact of regeneration and fragmentation in tropical habitats. And I think this really highlights the study of what you can gain from this kind of data set. And important insights for conservation as well. Some of them are actually very positive. And it's great to hear that regenerated forests can sustain high levels of diversity. I'm going to open the floor to questions. What I'm going to ask people is to please write their questions in the chat, and then I'll read them out loud. But I wanted to start with my own question. I obviously come from a molecular ecology background. So this relates to my personal interest. But I was wondering if you have any insights about the impact of these levels of regeneration or time post-regeneration on levels of within species genetic diversity, and whether you know if that responds as quickly as we saw that taxonomic diversity, functional diversity, and phylogenetic diversity responds.
No, not really, I'm afraid. So no, we haven't done, if I understood your question correctly, we haven't done any genetic studies, unfortunately. Weren't able to do so, weren't able to export the any samples yet. It's a difficult situation. in terms of exporting samples from Brazil. So unfortunately, no genetic studies. That is kind of, yeah, what I think would be nice to do, definitely, and would be a nice addition, or would have been a nice addition, yeah. So
watch this space then, you're saying.
Watch this space, yes.
Thanks,
the samples should still be there in Manaus, but yes, who knows, maybe one at some point we'll manage to do something with them.
I'll be very keen to hear about that.
Yeah.
We have a few questions from the audience as well. So we had a question about what about Myotis species and vampire and spectral bats, the effect of blood drinking bat species? Right.
So we've got, yes, we have vampires there as well. However, they didn't make up a large proportion of the, of our data at all. It's very difficult, obviously, to, and that's always the question when you, and the problem when you try to kind of then go down to species level, that, yeah, you might have, captured thousands of bats, but, then you have very few captures of the majority of species. That's how it is when you work in tropical environments, obviously. You have, one species that is super dominant. You've got a few common species, and then you've got this tremendous long tail of rare species. And although, yeah, we've got obviously vampire bats in our data set, but it's very difficult to do any decent and robust analysis based on a fairly reduced number of captures. That's unfortunately one of the key problems, I guess. Even working with a seemingly very large data set and some data that have been collected with tremendous effort and in the field over a number of years, but still, what I presented, for instance, in terms of this comparison, where we looked at also the seasonal effects and gender-specific effects on how species respond at the species level to local vegetation structure versus landscape structure. Well, in the end, we only ended up having enough data for 2 species, basically. So to do this analysis properly. So that is unfortunately of a limitation.
Thanks. And we have another question from Elise asking, which kind of variables are represented in vegetation structure? Shrub density, canopy height?
Yes, basically. So I've simplified that quite a bit and not gone into much detail there, but basically variables such as diameter of breast height, tree density, canopy height, canopy cover, vertical stratification and foliage density. And so they're kind of worked a number of variables together there in an ordination analysis to use that then in the modeling.
Okay, thanks. And we have a question also from Jack asking, have you managed to get any data or insights on the recovery of roosting sites within the secondary forest areas?
No, not really, unfortunately. But I think that is probably one of the reasons why, these gleaning insectivorous species are still not not kind of recovered entirely to continuous forest levels in secondary forest, because they lack often appropriate roosting resources.
I think that's a problem across, isn't it? In terms of the provision of roosting resources.
Yeah.
Obviously, the trees have to be a lot older in order to have all these characteristics enabled bats to roost there.
Yeah.
And we have a question from Sylph asking, how does the future of bats in secondary forests look like?
How does it look like? I mean, as I said, for secondary forests, to really work properly and function properly as an important reservoirs for bat diversity, we really need to make sure that they are adequately protected for sufficiently long to really ensure that they can maximize their or can fulfill their conservation potential to the most. I mean, it's the issue really is that a lot of them are really being cut down after a short time period, and obviously it will depend very much on the landscape, on the socioeconomic setting of a certain landscape. The BDFFP in many aspects is a kind of the best case scenario. Obviously, it's a protected area. There are basically very little other human influences there that you would commonly find in other landscapes, which are much more heavily impacted, obviously. So in general, the land use history also plays an enormous role. So there are a multitude of factors that will really determine whether secondary forest, actually will be able to sustain a considerable amount of bat diversity or not. But I think what is also very clear is that secondary forests, there are no substitutes for continuous primary forest.
Yeah, indeed. And we have time for one last question. So I have a question here from Thomas asking, are there any gaps in such research which you think needs to be further explored to gain a more comprehensive understanding of habitat fragmentation on community structure?
Well, I think in general, I think we need to, it's important that we have long-term data. I think that's really key, and we still lack good long-term data from more than just really a couple or a handful of sites. So that's the thing. One important thing that I think could, in terms of bat research, and if we want to understand how bats respond to landscape fragmentation in general, it would be great to have much more long-term data from a multitude of sites across the globe, basically. Also from, obviously other regions, Africa, for instance, and so on. I think that's very important. Also, one thing that I haven't shown or emphasized maybe in my presentation is we also found that there's a lot of natural variation also in continuous forests. So the bat assemblages in continuous forest also are very much, there's a lot of fluctuation in species abundances, and so really you can only get a good understanding of what's going on in these fragmented landscapes. First of all, if you have long-term data, and then if you have an appropriate study design as well, which allows you to kind of disentangle these large natural fluctuations from actual in continuous forests from actual fragmentation effects.
Yeah, I completely agree. And I really think that this kind of data that we need to understand the impacts of global environmental changes, we need these long-term ecological study sites like this one. And unfortunately, this is something that's quite rare, but definitely something that we need more data from these kind of experiments.
Yeah.
Okay, so I'm left with just asking everyone if we can thank Christoph again for this great talk. So if people want to turn their microphones on and maybe turn their cameras on as well, and just thank you again.
Hello.
And hope you all can join us as well for the next Bats Without Borders Wednesday webinar. And please do check our website for information about other activities that we're running.
Okay, thanks very much again for inviting me. It's been a great pleasure.
Brilliant. Thank you, Christopher. Great talk. And thank you everyone for joining in and asking such great questions.
Right.
Thank you very much. And I'm going to shoot off to Christoph. I'm going to drop you an email. I have a few questions myself.
Okay, fair enough.
Right, so Rachel, you finished the recording. I can close down now. Okay, brilliant.
Okay.